regards to an HIV vaccine, how is that looking in the future?
Well, currently it doesn't look very good. I was at one of the earliest international meetings on HIV that the vaccine issue had come up. They just found the virus. They were talking about an antibody test they were developing and they had developed, and so the first thing got there, we're going to have a vaccine for this in a couple of years.
Right.
So a guy gets up after that and said, well, you have to look at what a virus does inside the body to determine if that's even feasible. And he listed these 10 points, viruses that would make it easy to have a vaccine and viruses that would make it impossible to have a vaccine. HIV had all 10 of the ones that would make it impossible. And part of it is, very simply, is that when you get a vaccine, you're exposed to a dead virus or pieces of a virus, and your body develops what they call antibodies. So you learn to fight that and develop these antibodies against a certain level. When you're exposed to that, it'll kill it before it does anything. Right. Well, this thing, remember, the antibodies are outside of the cell. This thing enters the body often through a cell. So if somebody has sex and there's let's say there's these affected T lymphocytes inside let's say the vagina, it'll fuse with a T lymphocyte of the person that's not yet infected. Right. And then that'll enter the body through the mucosal membrane. Now, the virus is inside that cell. So all the antibodies in the world are not going to be able to even get into it if you have a vaccine. Yep. Now that cell will fuse with other cells, and other cells. Some of the cells eventually are going to break open and release more of the virus, and then the body can see it. And that's why the antibody response that you have, or somebody has that has never seen them, just got newly infected. Right. The antibody response takes several weeks, and but it's too late. I mean, you've already had all these cells that have been infected, so you can't get the antibodies in. So it's kind of like a Trojan horse. You've got this thing that enters your body, you can't see it, and by the time you do see it, it's too late. The virus is also a mutation machine, so it makes up to 9 mutations per application. It changes so much that the vaccine has no specific area that it can target. So it keeps changing. So like in the flu vaccine, it's only good for 1 year, and that works. And why? Because the flu virus keeps changing the coding. So the antibodies that you have against that influenza aren't going to help protect you the next year, most likely. So here you have to make a vaccine for every literal change that can occur on the surface of that virus. And that's almost impossible. That's again, but you still can't get those antibodies inside the cell to stop it. Right. Then you have these DNA that are reservoirs of the virus inside of a patient. And so maybe the antibodies might clear the virus in the periphery, which if it did, but you would still have the cells with the pieces of DNA in there that can make more virus. Right. So it just, you've got, there's so many factors against a vaccine. That's why, this was talked about as early as 85, I guess, or 84, 85. And to this day, we still don't have a vaccine that works. So it's, I've never really thought, even based on that very first lecture, that this was going to be something viable. Now, again, somebody come up with a brilliant idea and have some sort of T cell that may affect it, but that's, I don't know. Right. The cure thing is the same problem because you've got the DNA of the virus inside the DNA of the host. Mm-hmm. So I'll tell my patients, that even if you suppress the virus completely with the medications we have now, right, those cells will live for 70 years. So, I tell them, you take your medicines perfectly every day for the next 70 years, I can stop your medicines then and be cured. Most of my patients will be 100. So, that's silly. But we're finding ways to stimulate parts of that DNA. So in other words, let's say you suppress the virus completely. Well, one of the things I'm looking at for possible cure, that's not been proven yet, is that if you could turn on some of that DNA, so not the whole virus is produced, but some of the pieces of the virus, and those pieces can penetrate that suppressed cell, right, then the body can recognize that this cell is in fact infected, and it destroys it. So, it's like unmasking it. So,
Right.
so that reservoir of cells, instead of taking 70 years to basically die off naturally, we might be able to enhance the destruction of those cells much quicker. 4 years, 5 years, 10 years, we don't know. But that's one of the approaches they're taking as a possible cure. They have done the cures with the bone marrow transplants. There's only 2 people that have been cured like that. It's $1 million, and there's been quite a few people that have also had it and they've died from it. So
Right.
it's not a practical cure.